Hello, ladies and gentlemen, and welcome to today's broadcast on challenges of establishing laboratory-developed tests by Dr. Vijay Singh. I'm Bill Anliker from Thermo Fisher Scientific, and I'll be moderating today's event. Before we begin, I would like to encourage you to engage with us. You can submit as many questions as you want at any time you want during the presentation. Simply type your question in the Q&A box on the left-hand side of your presentation screen and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Please welcome Vijay.
Hello, everyone. Today I'll be talking about our experience here at HealthTrackRx. We are a specialty infectious disease testing lab right outside Dallas. And so I'll be talking about our experience in establishing laboratory-developed tests, or LDTs, specifically for disease diagnostics. The presentation today will be covering, in very broad terms, the three basic questions that basically define anything in life: the what, why, and how of establishing an LDT in your lab. So what is an LDT? As the FDA defines it, now technically an LDT is not FDA approved and is defined as an in vitro diagnostic test that is developed, validated, and commercialized for use within a single laboratory. And for anyone who's interested in developing LDTs for use in their lab, I would highly recommend the website that you see on the slide right now to visit this website, because it has some really great information of what defines an LDT and how to get started. So now the question is, why are LDTs needed? Now, very often, and I'll show you some examples later, it is the case that certain diagnostic tests are not available commercially, and there can be a number of reasons for that. Maybe it's a rare target that you're interested in, or the clinical utility of that particular test is not very well defined, and it is simply not commercially viable to bring those tests to market. Keep in mind that developing and getting full FDA approval for a single test, for the manufacturer, it can cost upwards of $10 million, to get full FDA approved test, and sometimes you'll find that the FDA approved test that is available on the market, its performance is inferior as compared to some of the other LDTs that are running. And the amount of flexibility that one has in establishing an LDT with respect to using multiple manufacturers and sources that will bring together that test, that altogether sometimes makes an LDT both attractive and necessary. So now that you have decided to bring the pain of establishing an LDT into your life, how does one go about doing that? And the very first thing that you do is that you look for, the regulations and the guidelines that define this process. And this is the very point that it begins to, get confusing, because you have all these very different regulating and guiding authorities that define both the test characteristics and the laboratory practices. Now, within the US, the ultimate authority on LDTs is the FDA, or the Food and Drug Administration. However, what the FDA does is that it practices enforcement discretion. And because there is clear, and I'll be talking about that in greater detail in the next few slides, and individual state laws that regulate the functioning of the labs, and the FDA lets these regulating bodies control the tests that are being run in the lab. However, once you are getting into the process of developing an LDT, what you'll realize is that all of these regulating authorities or guiding authorities have certain set guidelines, and you'll find there is a major overlap in these guidelines, and any of the resources that are provided by these authorities can be a good starting point. So here in the US, it's the Clinical Laboratory Improvement Amendments, or CLIA, which was established in 1988, that regulates all testing of human samples in a laboratory. And these guidelines underwent extensive revisions in 2003, which is now called the CLIA Final Rule. And it defines the various test performance characteristics that need to be established for bringing an LDT on board. But what you should also keep in mind that depending on where you are located, you may have more extensive and stringent local state laws. So one should also keep that in mind while trying to develop these tests. Now, here is, very broadly speaking, the different test performance characteristics that are defined by the 2003 final rule. Right? We're talking about the reportable range, the analytical sensitivity and specificity of the test, the precision of that test on a day-to-day or over a time period. Because remember, you can be precisely wrong about something. The accuracy of the test and the reference intervals. Now, I'm not going to get into the technical details, as these guidelines are widely and very clearly available, and that would entail a whole different presentation. But the important point that one has to keep in mind is that according to CLIA, one has to establish, now, albeit not in the same great detail, but you have to establish these test performance characteristics even for FDA-approved tests in your laboratory. Okay. So what was our experience here at HealthTrackRx in developing some of these LDTs? It was somewhere towards the end of 2017 when we launched our large multiplex syndromic real-time PCR-based test panels that cover a wide variety of tests with respect to respiratory, urinary tract, sexually transmitted infections, gastrointestinal, and wound, dermatology, and ophthalmology based infections. And this was our guiding principle, that what we wanted to do was to develop a testing strategy that incorporated a multiplexed, real-time PCR-based syndromic panel approach that saved both time and money, again, for both the patients and their providers, that ultimately results in better clinical decisions with respect to patient care. So in brief, the idea was to provide the maximum amount of clinically relevant information to both the patient and the provider, and to do so at a reasonable cost, again, to the patient and to us at a reasonable cost and profit margin. So to anyone who's thinking about establishing LDTs in your lab, right, the first thing that you should do is that you need to determine a testing strategy that works best for you, not only as a scientific exercise, and I'll be, going into some details on that, again, not just as a scientific exercise, but as a business decision also. And it is not just the test or the assay alone that has to be the focus. It is the whole workflow within the lab that feeds into that test that needs to be streamlined in such a manner that the wastage of time, money, and resources is minimized, and the information that is generated for both the provider and the patient is maximized. And again, I'll be bringing up this point again and again, is that as you begin to think about developing these strategies, these workflows, or bringing these tests on board, right, you would want to work very closely with your assay manufacturer or your test developers, right? So what was our approach when we looked at the whole workflow, right? Here is an example of our respiratory infection panel, or the RPP panel. And when you look at these targets, I can tell you with a very high degree of confidence that if you go about looking for FDA approved tests on these, there are maybe one or two non-viral targets that are FDA approved. And depending on the manufacturer that you may want to work with, uh, you may have anywhere from 2 to 6 or maybe 7 viral targets that are that have FDA approved tests available. And when you looked at the very extensive list of antibiotic resistance markers that we have, only 2 of those targets are really reimbursed by the payers, right? So if you would go about looking for, again, FDA approved or CLIA waived tests, such an extensive list is simply not available. However, what we've seen in a number of cases, there is a, a huge amount of clinically relevant information with respect to, the pathogens or the infections that might be present in a patient sample that would simply be missed by a lot of these point of care tests or FDA approved tests. And we published a paper in 2019 detailing, the severe limitation of some of these point of care tests up to 50% of the time, where clinically relevant infections would be missed by some of these tests. And so that in itself validated our approach towards developing the LDTs. And to talk about the workflow, as I said, everything else that flows into that assay is also equally important. So we spent a significant amount of time and resources in developing a lysis and transport medium. And I can say that we've sort of achieved true synergy, wherein we use this single medium for all our different sample types. So whether it's RPP, STI, wound, GI, whatever the sample type may be, we use this single medium for transport, and that allows for in-transit lysis also. And again, like I said, as a business decision, when we looked at developing the LDTs, what we realized that that running these extensive panels would make sense at a certain patient volume or sample volume. And for that, it became very, very apparent very soon is that we would need automation at different levels of the workflow. And one of that point, which was proving to be a big bottleneck, was the extraction of the nucleic acid from the samples. So we spent, again, a significant amount of time in developing automated processes for the extraction, and which is, once we have the nucleic acid in hand, we follow it up with reverse transcription, pre-amplification of the samples, because we found, or what we ultimately went with, was a nanofluidic real-time PCR-based platform, which allowed for really high throughput performance of the PCR, and then followed by that real-time PCR that I was talking about, ultimately leading to the report, which is the product that we put out for the providers and the patient. Again, we have streamlined this workflow, which again minimizes any wastage with respect to time and resources for all our different sample types. Okay. looking at some of the examples with respect to the performance characteristics. And herein, I would emphasize the point of working closely with your assay manufacturers. Is that one of the biggest stumbling blocks that labs face in developing any extensive panels is finding the reference material of you know wherein you'll use that reference material to establish some of those test characteristics. And it can be very, very difficult to find true relevant reference material. So the manufacturer that we worked with was able to provide us in a timely fashion, all of our target sequences at a defined known value. Okay. What that allowed us to do was to establish the limit of detection of our assays and to basically do good dilution assays that allowed us to look at a good linear correlation for all of our assay targets. So you can see what we received was 10 raised to power 5 copies of our target sequences, which we could very quickly do a good dilution assay and see very, very good sensitivity for the test. And in the meantime, we kept working with a lot of sources, so public health departments, hospitals, clinics, in sourcing true known patient samples with clearly identified infections. And some of the relevant examples are here. And you can see that we could establish very good linear correlation for all of our assays. And that allowed us to, define a lot of our test characteristics that are laid down by the different guidelines, whether they be from CLIA, CAP, or ISO, or the FDA. And now, I'll be talking about a more, timely example, which was the establishing of a COVID-19 test here at HealthTrackRx. And it was somewhere in February of 2020 when the COVID-19 was declared as a public health emergency and as a pandemic. And so we moved very fast. Now, keep in mind that our experience was more with these multiplexed large panels. And we never, till this point, we haven't really done any single target, single pathogen type PCR tests in the lab. But we moved fairly quickly in trying to establish this test. And when we started generating data for this test in the lab, the emergency use authorization that was given by the FDA was very limited. It was only for a particular extraction type. It was only on a particular machine, the ABI 7500, which we did not have in the lab. And so what the FDA suggested was that if you wanted to do anything else, they defined it as a bridging study because they had laid down the test guidelines or the characteristics or the performance characteristics of these tests. And so we started with defining all the various different sample types, again, the automated RNA extraction and the real-time PCR performance on machines that were frankly different. And we didn't have any resources at that moment of getting any of those machines in the lab. And frankly, we did not have the time to validate those new machines. Okay. And then the assay was also different from what was approved at that point. And again, the different data points and different test characteristics were established. So here is our whole table for the limit of detection. And you can see that it was established at 10 raised to power 1 copies of the viral target per microliter. And it's not that we could not detect it at any lower values. If you would go, that we went right down to 10 raised to power 0.1 copies per microliter of our target. However, we made a decision of capping the limit of our test at 35 CT values, because if you look at the linear equations, even though this is great linear correlation for the different assays or the different targets, you can see that, if you look at the equations themselves, the moment you start getting beyond the 35 CT value, uh, the clear determination of whether the sample is positive or negative would become questionable. And then we had all of our samples from the 2019 study saved in a deep freezer, and that allowed us to do a good specificity study wherein, all the other syndromic targets, we found that there was no interference from those. And overall, the specificity of testing a difficult sample type like the sputum samples as opposed to a nasal or an oropharyngeal swab, put together, the predictive value of our test, both positive and negative, was found to be 100%. And then at the same time, we moved very quickly to do interlab correlations. And, for as per FDA guidelines, we took the difficult sample type, which was the sputum, and we correlated our results with another lab. And that allowed us, for the granting of our EUA. And since that time, I would say in the last 18 months or so, we have performed nearly 2 million COVID tests. And so why I gave this particular example was that when we were generating this data with respect to filing for the EUA application, we were also following all the guidelines from with respect to a inspection. And the idea was that all the technical repetitions, all the biological repetitions, all the precision studies, the accuracy studies that are needed, that we had all of that data on file for our CAP inspection. And when our CAP inspection window opened in February of this year, we passed it with flying colors because we already had that data. And, for people who may find it difficult, and again, yes, we were coming from a place of high experience, but at the same time, this was a new approach for us. But all of this work, if one thinks that it is difficult, yes, it is. However, if you're planning, and as I used to say in a previous life to all of my students, that 1 month on the bench will save you 2 hours in the library, it's an excruciating amount of planning that goes into it. But all of this work with respect to establishing the COVID assay was done in a period of 10 days. So with that, I'll summarize this presentation that yes, establishing an LDT can be a very daunting task. And if you look at the different regulating bodies, and keeping in mind that you may have, particular state laws also governing you, this can be very, very confusing. However, if you look at those guidelines in a very broad sense, these are the basic principles of good laboratory practice that are guiding you. So follow those guidelines, and follow those guidelines to a T, keeping in mind that you will have to pass an inspection, right? And if you're doing that, again, there can be limitations with respect to resources and what your testing strategy has to be, what your business strategy is, right? You will have to make those decisions. However, the decision of establishing an LDT in the lab should always keep the patient interest at its core, right? What is the clinical utility of the test? How will that test impact the patient life? How will that test provide all the clinically relevant information to the provider to make the best decisions with respect to the patient's care, right? That has to be kept in mind, right? After that, right, what you should always do, you know, you have to remember that this ultimately is a business decision. Okay, so you will have to bring all of that together, and the workflow in the lab, and the costs of running that test, and what would be the reimbursements. All of those different things have to come together for you to make that decision. And again, I cannot emphasize this enough. If you ask nicely and work closely with the manufacturers of those assays, it can be a very easy task, because like I said before, you may be resource limited, but realize this, that people and collaborators around you will have expertise that you may not possess at that time, and they will help you bring together these LDTs, you know, to the market, and to your patients, to your providers. And it can be, a task that is easier than it looks. And with that, I would like to thank the whole infectious disease team here at HealthTrackRx. Uh, Again, I'm privileged to work with such a great group of people. In the last 1.5 years with the pandemic, I've never seen, again, a more committed group of people. They've been through hell and back, and I see them very tired all the time, but never wavering in their commitment to the patients and to the turnaround time. And I would like to thank all of you for listening, and I'll take any questions that you may have. Thank you.
Thank you, Vijay, for sharing your personal experiences and reflections of what it takes for a clinical lab to bring a in-house molecular test to market. We have received some great questions already. Vijay, what advice would you give to a laboratory looking to implement its first LDT?
Again, great points to emphasize again. You know, the first and the foremost that they should look for, again, like I said, there are some great FDA approved tests available, right? And they have to keep in mind that what is the niche that they are targeting. Is it clinics? Is it long-term care facilities, hospitals, or OPD population? And once you have that clarity, and you know what sort of sample volume you would be looking at, and then what the market needs are, where the gaps are, and they want to fulfill those gaps, then they need to define the testing strategy that would go. And again, it has to be with the realization that some of the time and the resource that needs to be put into developing these LDTs can be fairly extensive. It can be expensive for a lot of labs. So first, clearly define the need that you want to fulfill, and then look at the resources that are available to you. And again, collaborate. Like I said, one of the big stumbling blocks in defining the test characteristics is finding the resource or the reference material. So work with the public health departments in your area, work with hospitals, connect with the universities around you, and clinics around you. And like I said, work very closely with your assay developers if you're not doing it in-house. And you know all of those resources coming together, all of those expertise besides your own coming together, will will make this whole exercise easier.
Great. We have a two-part question. It reads, do you have any tips for someone who is new or unfamiliar with panel development and testing? And are there any resources you find particularly helpful?
So, like I said, that has to be, extensive planning. And I can talk from our own experience here. Is that you need to, one, you can do a survey of the clinicians around you, and they will tell you what sort of tests they are interested in. Do a extensive literature search. The idea with us was to bring together, a very high percentile of the pathogens representing each syndrome. And then obviously at the same time, we did a market survey of the clinicians around us. And, they pointed out certain targets that were not a part of our original list, but we found that there was enough interest in the clinicians for those particular tests or for those particular pathogens for each panel type. And again, I say this with the full realization that not all laboratories are going to have those resources at their disposal, or they may not have the time or the money to invest into that. However, I would still very seriously suggest for everyone to try and take that approach, because in the long term, that would allow you to put a good product out in the market like I said, I keep saying again, for both the patient and the provider. And then how you design your workflow will allow you to do it at a price point that would, again, maximize the information and minimize the cost.
Vijay, how quickly are you able to change your panel content and bring a new test to offer to your clients?
So the nature of our large multiplex panels, the way they are engineered and the way the technology is, it takes anywhere from 4 to 8 weeks if we have to make extensive changes on our panels. However, depending on, I'll give you an example. What we realized with our STI panels was that a large number of the providers were interested in gonorrhea, chlamydia, and trichomonas test only. And so what we did was that we created a small panel specifically for that. And we basically took these 3 targets out of our large multiplex panel. And again, I think we'd already cut our teeth with the COVID assay. And from the standpoint of manufacturing, it took less than 3 weeks to do that.
What would you say is the single most important thing to keep in mind when developing a successful test?
Personally, I would say that you need to look at the clinical utility of that test, and always keeping the patient's interest at heart, but not losing the perspective that yes, it is a business decision also. So one has to bring in different aspects. There is no one true fact or aspect that defines the LDT. So you will have to keep in mind that is there, a gap in what is available to the patients? Is there a certain market need? And again, if you do decide to bring that on board, what would be the implications of running that test with respect to payer reimbursements? And even if those reimbursements are low, can you design a workflow that will minimize your cost and would allow you, depending on the sample volume, would allow you to perform that test? And can you bring that, or can you commercialize that test within a reasonable profit margin? So all of those aspects truly come together. And so I wouldn't say that there is one true aspect that is overarching and defines it.
We do have time for one more question. Vijay, what are the most time-consuming or challenging parts of the process for bringing a test to market? Do you have any advice?
So, like I said, one of the, it's the actual planning of the assay itself. And, once you have identified the system that you want to work with, and then, there are certain areas that are more clearly defined. Like I would say in the whole process, the nucleic acid extraction process, that's more well defined. There are certain known ways that in which you do it, and you may have experience with one system versus the other, and those are easier still. However, the design of the panel itself, you know, and then depending on who your manufacturer is, that can take a long amount of time. And then once you have the assays in-house, once you have them in your hand, the thing that can take a bigger amount of time is finding the proper reference material. So I would suggest that one should start looking for the certified reference material, whether it's through, a known source like ATCC, which can give you uh, the pathogens themselves, and you can spike the proper matrix and do those tests, or you work with synthetic pieces of DNA, or you work with hospitals or public health departments that can provide you with those samples, or work closely with the universities in your area that may have some research material available to them. But that is one aspect that takes a, a long time to bring together. And then again, it's the planning of those experiments. So read those guidelines carefully, and they very clearly define the basic minimum criteria of doing each experiment, and what are the repetitions and what sort of statistics need to be done on that data. They clearly define those. So make sure that your planning of those experiments is done, beforehand. Again, follow those good laboratory practices, and it'll be a much easier task.
Thank you, Vijay. That is all the time we have for today. As a reminder to our audience, questions we did not have time for today and those that are submitted during the on-demand period will be addressed via the email address you provided at the time of registration. So thank you again to our speaker, Vijay Singh, and to everyone who participated in this presentation. We hope you enjoy the rest of the event, and please be sure to check back frequently for new content and presentations added throughout the year.